starting our technical session. Dr. Balaji, please take over the mic to coordinate the technical session. Our chair and co-chairperson have been seated. And Dr. Jacob Philip Matthew, I hope he's on the line. Okay.
Good afternoon. Yes, I know it's slightly difficult to keep awake after a very good lunch, but still we'll try our level best to keep you awake and listen to this session. The, the second, it is in the hands of the speakers now. Okay. The second session will be having 3 lectures. One is by Dr. Jacob Matthew, second is by Dr. Gurudev Singh, and third is by Dr. Balasubramaniam Vairiyappan. The chair and co-chair for this session will be Dr. Venkatesh Devanur and Dr. Pushpavalli. I would request the chair to conduct the sessions now. And the total time allocated for each section will be around 20 minutes with the Q&A session included in that. Thank you.
Thank you, Dr. Balaji. Welcome, Dr. Jacob Matthew Philip. We have today post-lunch session. I'm sure people are quite awake. It all depends on Dr. Jacob how to make us more awake also. Thank you. And the subject is NEEM in dentistry. NEEM in dentistry is an age-old Indian DNA. Every village, datung, they take in the morning and brush it. So NEEM in dentistry has been a part of Indian DNA. And now, scientific outlook, benefits, current status, future direction, from a very scientific, methodical point of view, Dr. Jacob Matthew, he's from Muscat, and he's an orthodontist. He's giving an online presentation on this. Over to you, Dr. Jacob Matthew Philip. Thank you.
Good afternoon. Thank you very much for those kind words of introduction. And it is my immense privilege this afternoon to be able to share a few insights about NEEM in dentistry. And first of all, I would like to congratulate the organizing team members, the World NEEM organization, for organizing this platform to promote the use of NEEM. And a special thanks to Dr. Nirmala for having extended this invitation to me. And it was a great pleasure to listen to the earlier lectures, and congratulations to the golden couple for having set up a neem forest. It's almost unbelievable, but thank you for showing us the way. And also Dr. Jayashri, who was able to enlighten us about the uses of neem as an antiviral and also against cancer cells. And thank you very much for this invitation. Now I would like to begin my presentation on uses of neem in dentistry. As you all know that neem has been used in traditional Indian medicine, and it is our job as scientists and researchers today to be able to provide enough evidence to be able to bring this into modern medical practice. Why do we have to use neem in dentistry? Well, if you would ask me, there are many reasons why neem can be used in dentistry. First, neem can be used to mitigate antimicrobial resistance. As you all know, nowadays there is a lot of resistant strains that are on the rise, and the neem can be used to support good antibiotic stewardship, especially in the field of dentistry. Secondly, I would say that it is able to provide an accessible and affordable healthcare for us, especially if you're using it at a preventive stage. Then it can reduce cost and reduce disease burden. And thirdly, it is a culturally accepted botanical. And so if you're going to use neem in medicine, we can definitely show the patient compliance and there will be a significant improvement in health. As we were hearing from the lectures this morning, there are a lot of phytochemicals and bioactive compounds in neem, and they are available from all parts of the tree. And there are various mechanisms by which this neem is able to give us the medicinal effects. For example, the terpenoids, limonoids, flavonoids, and tannins They're able to inhibit certain enzymes like cyclooxygenase, lipoxygenase, and matrix metalloproteinases through which they are useful in healthcare. And polyphenols and phenolic acids, they are used in health. They can be used in healthcare because they have a mechanism by which they are able to inhibit enzymes that are related to a protein called as an adenosine protein, thereby reducing biofilm formation. And the supponents also reduce oxidative stress and reduce enzymes like myeloperoxases, and they are also useful for reducing inflammation. And other components like alkaloids are helpful in promoting wound healing because they modulate enzymes for wound healing and fibroblast growth factors, collagen synthase. So these are the ways in which they are able to have a mechanism where we can utilize these mechanisms for the benefit of human health. Now, there are various domains in dentistry where neem can be utilized. One is for maintaining oral hygiene. So, as you all know, neem toothpaste and neem mouthwashes are available and they can be used in reducing bleeding from the gums, reducing plaque and gingival scores, and reducing even bad breath, mouth malodor. And in the field of periodontology, that is the field concerned with the gums and supporting tissues of the tooth, they're useful in inhibiting periodontal pathogens and preventing gum disease and gum loosening. In my specialty, the field of prosthodontics, we can propose the use of neem as a denture cleansing solution because I feel that neem is able to prevent the formation of biofilm, especially on the denture surface, and it can be used as an adjunct in prevention or treatment of denture stomatitis. I will be speaking more about this topic for the remainder of my presentation. And the field of endodontics, neem has been tested to be used as an irrigant to reduce the bacterial load in the root canal. And also it is used as an intervisit intracanal medicament. And it has also been shown to be effective against E. fecalis, which is a bacteria associated with recurrent root canal infections. In the field of oral surgery, it can be used to promote wound healing. It reduces edema, reduces inflammatory cell infiltrate, promotes epithelialization and collagen formation. And more recently, in the field of implantology, it has been used as a coating around dental implants to reduce biofilm formation. As we were hearing this morning from some of the delegates, there are various safety and toxicology considerations which need more research to be addressed. The route-dependent safety profile, the dosing considerations, adverse effects, and the contraindications. And standardization is a challenge. The batch-to-batch variability, the variability of the active components being extracted, the quality control. These are all aspects that need more research to address. And the use of NEEM is well aligned with the United Nations Sustainable Development Goals, especially with the goal of good human health and well-being. And I feel that the World NEEM Organization is strategically poised today to promote the scientific validation of NEEM and to provide a platform for fostering partnerships between the various sectors like policy makers, industrialists, academics, and researchers so that we will be able to bring in NEEM more and more into our lifestyle and promote health. Well, there are various gaps and challenges that we need to face. There are gaps in evidence base. So scientific research would be able to address this evidence based gaps. And there are standardization challenges, then production challenges, and how do we translate the research into our day to day life. All these are various gaps and challenges that we need to address. And so, as a dentist and belonging to the field of prosthodontics, I looked how are we able to use NEEM in dentistry? What is the problem, specific problem? What is the lacuna that is existing? And so I came across this particular problem called denture stomatitis. And some of you would know about this disease. It is a fungal disease of the oral cavity caused by a fungus called Candida albicans. And Candida is a commensal in the oral cavity. It is always present in everybody's mouth. And during certain conditions, like when the patient's immune status is reduced, for example, long term steroid users or those who are having some malignancies, those are undergoing radiation therapy, cancer chemotherapy, and such patients who are old or malnourished are immunocompromised. then in those patients, this commensal in the mouth can turn pathogenic. So what happens is this Candida fungus attaches onto the surface of the denture, and then when the patient wears the denture, they get an oral condition called denture stomatitis or Candida associated sore mouth. And this condition is actually seen in patients who are unable to maintain denture hygiene. Now, many methods, sorry, yeah, many methods have been used to maintain denture hygiene. For example, the denture toothbrush. But when patients who are wearing dentures are already old and with limited manual dexterity, they are unable to maintain denture hygiene well. And other methods of maintaining denture hygiene have also been looked into, like the chemical methods and physical methods like microwave radiation. But all these have been shown to affect the physical property of the denture. And so very often at the end of the day, we resort to prescription of antifungal medication to treat this condition. And we see a rise in antimicrobial resistance, especially to the azole group of drugs, fluconazole and miconazole that we use for treating this condition. And so we want to look at an alternative that can help us in the management of this condition. So this is the problem that I have identified in my practice. And so when we look to nature for help in this time, we see we can look at the neem leaf, the humble neem tree, and it is considered as a safer alternative, which is environmentally friendly. And we already know that it has antiseptic properties from previous research. And so the objective of my study was to evaluate the antifungal effect of the neem leaf extract on the azole resistant and azole susceptible strain of Candida albicans. And so we went about preparing the neem extract by drying the neem leaf and then powdering it, dissolving it in the solvents available, ethanol, ethyl alcohol, methanol, acetone, chloroform, and water. We put it in an incubator shaker and dried it. And then we obtained the extract, and then we mixed it with the DMSO and stored it for use in the future. And then we subjected this to two further examination, the agar well diffusion assay and microbrod dilution assay. So we took the Muller-Hinton agar and we tested the extract along with the controls amphotericin and saline on the different strains of the fungus. And we also did a microbrod dilution assay where we used a serial dilution of the neem extracts, and then we checked the minimum concentration which was able to inhibit a spot inoculum. And then we came across the results which were very interesting. In fact, the agar well diffusion assay found that the positive control was giving a 15 millimeter zone of inhibition, whereas the ethanolic extract of the neem leaf was able to give a higher zone of inhibition, 16 mm for the azole resistant and sensitive strain of the fungus, and 17 mm for the standard MTCC strain of the fungus. So antifungal efficacy of the ethanolic extract of the neem leaf was comparable even to drugs like amphotericin B. And the other test that we did, the minimal image concentration for the various extracts, we found that ethanol was the extract that was able to give provide antifungal efficacy for all three strains of the fungus, the susceptible strain, the resistant strain, and the standard strain. And the MIC value was found to be 50 microgram. Now that we have established the antifungal efficacy of the ethanolic extract of neem leaf, we wanted to check its effect on the denture base material. So the denture base is actually made up of a resin, and we wanted to make discs of this resin. So we went about making small discs using the compression molding technique, and we made these discs and then used it for two further tests. One is the fungal adhesion assay. So we wanted to find out how well the fungus is adhering onto the surface of this disc once it is treated with the plant extract. So for this, we took the discs and we incubated it. We pre-treated it with this plant extract and the gold standard, which is the chlorhexidine antiseptic, and normal saline. And after washing it, then we incubated in the yeast cell suspension, and then we washed it and did a staining and yeast counting. And the results were again surprising. We found that if you look at this diagram, the blue color bar is the mean yeast cells adhering onto the denture base, which were pre-treated with the neem leaf extract. And when compared to the gray bar, which was the disc treated with the normal saline, we see a good reduction in the adhesion of the fungus on the denture base material. You can also see from this diagram the yellow bars which signify the mean E cells adhering to the denture, which was pre-treated with the chlorhexidine, which is the gold standard. Of course, chlorhexidine performed better, but we also see that the extract of neem leaf was also able to give significant reduction in the addition to the denture-based resin. And we see that this reduction in addition is also consistent. It is able to consistently inhibit the reduction, the addition of the fungus on the denture at 3 hours and 36 hours of incubation. So even across a period of time, it was able to inhibit the fungus from adhering onto the surface of the denture. So very good statistical results we were able to get. And second test we did was the antifungal efficacy. Now, addition of the fungus on the denture base is one, but how much of that fungus is actually viable and how much is dead? We wanted to find that out. So we did the second set of tests where we took these denture base resin discs and then we pre-treated it with the plant extract with the chlorhexidine antiseptic and saline, and then we incubated it in the yeast suspension, washed it. Then we did ultrasonification and took the sonicated solution, serially diluted it, plated and incubated, and then we check the colony forming units. And these are the results that we obtained. You can see the red bars are the bars for the drug azole resistant strain of the fungus. And we can see that the neem leaf was able to definitely reduce the viability of the fungus as compared to the chlorhexidine antiseptic. And this shows extremely promising results. And the blue and the white bar are the amount of viability for the resins we pre-treated with the extract, and we can see almost zero. They are able to completely destroy the fungus from which are adhering onto the resin. We also wanted to do some preliminary biocompatibility testing. What is the effect of this extract on cells? And preliminarily, we used the mouse fibroblast cells for the MTC assay, and we obtained the elute of this disc, which was pre-treated with the extract, the neem extract, and we used it to do this MTT assay, and we read the absorption, and we see that the neem leaf extract was able to give better viability results when compared to the chlorhexidine. So it is from these results, it is more biocompatible from the chlorhexidine antiseptic mouthwashes. Also, as I previously mentioned, the other experimental methods of cleaning and sterilizing the denture-based resin had deleterious effect on the property of the denture material, significantly weakening the denture material. Now, we wanted to check whether the neem leaf extract had any such effect on this resin. So we made these bars of the denture material, and then we immersed them for 30 minutes every day for 30 days in the plant extract, and then we subjected it to the universal testing machine 3-point bend test to check the flexural strength. And we can see the results in the graph here. There is no significant difference in the flexural strength of the material even after treating with the neem leaf extract. We further went on to identify what are the components that are present in this extract that we have got, and we did the GCMS and FTIR, and we wanted to check about how it is going to interact with the fungus. And so when I looked at the literature, I found that most of these antifungal drugs are targeting this particular receptor in the fungus, Candida albicans, called N-myristoyl transferase, which facilitates a particular enzymatic reaction which is essential for the survival of the fungus. And this is the antifungal drug that is commonly targeted in the antifungals. And we wanted to check the interaction of the compounds that we have identified from the ethanolic extract of neem. And we carried out the molecular docking using the various softwares, orthodox software that is available. And we found out the binding energies, and we saw that we got very promising results with many of the extracts. So this is in brief about the study that was performed regarding the use of neem in dentistry. And in conclusion, what we found that many of the components of the neem extract is able to act on the fungus and prevent its primary addition on the denture base. And not only that, it is able to reduce the viability of the cells that are already adherent on the denture base resin. We also found from preliminary investigation that it seems that it is safe to be used with the mouse fibroblast cells. Of course, further testing is required. It was also found that it was not affecting the flexural strength of the material, And preliminary docking studies also we have done, and we were able to see that many of the components had a very good docking interaction with antifungal targets. Of course, we need to, much more result is required. This is very a preliminary study. We have to see how we can use this in clinical trials, how to make it into a translational research so that it is actually used. And we should also do more research to find out how we can use this particular extract in preparing denture cleansing solutions, and then how we can market it, so that we see that many of the denture wearers who are old, and 25 to 65 percentage of prevalence of this particular disease is there in denture virus, whether they are aware of it or not. And it can also spread from local to a systemic spread. So this particular area has to be focused on in future research. So thank you very much. And this is a photo of some neem trees in Muscat, Oman. And incidentally, I also wanted to mention Dr. Nirmala had written a news item which was published in the Times of Oman more than a decade ago, and she had so nicely shared it with me. So I was so happy to be part of this community that is promoting the use of neem in healthcare. So thank you very much for this opportunity. It is our a responsibility as academicians and researchers and industrialists to use this traditional knowledge that has been there in our country, and and bring it to modern medicine and bring it towards modern practice, and then we can create a more sustainable healthcare. Thank you very much for this opportunity. I hope it was useful for you. Thank you for this opportunity.
Thank you, Dr. Philip. Perfect timing, exactly maintain the time and interesting topic for everybody. Hope everything is safe with you and everybody in Muscat, Oman. I wish all
Yes,
of you,
unfortunately, I wanted to be physically present there, but because of the
I know,
situation, I couldn't fly.
I
Thanks,
know, but
sorry.
hopefully at least next session, next meeting, we'll be able to have a personal meeting with you. It's been a very
Hopefully,
interesting
yes.
thing. We have some 5 minutes of time for anyone if you want to have any questions to Dr. Matthew. We have another dentist, Dr. Mahak is there. She's helping. She's the MC and producer of all YouTube films on NEEM. She's also a dentist, your community. Yes, doctor.
Hello, sir. Wonderful work. I would like
Good
to
afternoon.
compliment you. Wonderful, wonderful work. Is it possible for us to be a part of your team and help you with your research in any way?
Yes, definitely. As I had told you, this research that has done is quite preliminary. So we need to do clinical trials,
Okay.
and we need to see how we can make a product out of this. As I was talking to you about the denture cleansing solution, so that has been on my mind. So we need to work as a team, and because I am only the clinical part of the team, so I need somebody else to that I can collaborate with, and then we need to see how we can make clinical trials and then make a product like a denture cleansing solution, which can be useful for denture bearers, especially those who are immunocompromised, who are susceptible to this particular fungal disease of the mouth.
Yes, because I already have a patient, he's a cancer patient, and chlorhexidine solution is not able to help him with stomatitis. It's causing another
Yes,
kind of reactions in his body. So we were actually looking for a natural solution because he's a big believer in Ayurveda. So
yes.
we are hoping to get in touch with your team so that we can do something about it, as he's not able to wear his denture properly because of his
Yes,
cancer
this
treatment.
is exactly the problem that I have been coming across. Patients who are, even after the antifungal standard treatments are being given, the strains are resistant, and so the oral lesions are not healing. So that
Yes.
is why I was looking whether we can use the neem leaf extract as a therapy for these patients. And through this research, I have found that it is active against the azole resistant strain of the fungus. So this is has a promising result, but of course this is preliminary work. As I already mentioned, we need to do clinical trials. We need to see how we can formulate a product as a denture cleansing solution from here.
Thank you, sir. We would love to get
Yeah.
in touch with you, and if we can help you in any way, it will be our pleasure. Thank you, sir.
By all means, my email address is there on the last slide, so you can always mail me. We can
Sure.
see how we can take this forward.
Sure, sir. Thank you so much.
Thank you, doctor. And we have
I just
definitely.
wanted to make a comment to you that I'm from the Bahamas, And my dentist in the Bahamas did a trial. 5 out of 6 of her patients, she used the neem leaf extract for post extractions for irrigating
Yes.
the cavity, and 5 out of 6 were positive, no infection, nothing. The one that failed was a smoker and didn't do any of the post treatment that was recommended. And the other thing is that the neem oil has been very successful treating gum disease as well. For when you have an abscess, using the neem oil directly on the gums has helped to prevent having to have deep surgery. Just a comment.
That's wonderful. So the neem is actually acting on the collagen synthase enzyme and also promotes fibroblast growth factor, and that is the reason and essence why it is able to promote wounds in the oral cavity and ulcers. So that is fantastic research. Thank you for sharing that with me.
One more. Yeah, Doctor.
Curiosity. Uh, there, why is there a difference between the azole sensitive and the azole resistant strain? There is a difference in the activity of neem leaf extract, isn't it? So what is the reason between, you know?
Yes, from the study that I had
Yes,
conducted,
yeah.
the difference in the adhesion of the fungus to the denture resin, which was pre-treated with the neem leaf extract, was not uniform. There was a difference between the 3 strains. We had used 3 strains of the fungus. So one was the MPCC 3018 strain, which was a standard strain, and then 2 were strains which we had isolated from a patient who had the disease. So one group, one strain was actually resistant to the azole drug, and the other was sensitive to the azole drug. So we used 3 strains of the fungus, and we had these denture resin discs which were actually immersed in the plant extract and then exposed to these fungus, and we wanted to check how much of the fungus is actually adhering onto it and how much is viable at the end. And we saw that this is not uniform. There is a difference in the amount of fungus that is adhering to it and viable. We do not know the reason. There is more tests to be conducted, but we did see that there is a difference between the strains used and the amount of addition to the denture base.
So,
But we do not know the reason.
okay. And my other
We need to
question is,
investigate further.
And my other question is, your cyclohexidine results and the NEEM results, there is a significant difference,
Reduction, yes.
reduction, but not to the level that,
No.
it is expected. So
Yeah.
a combination of cyclohexidine and a neem extract together as a formulation can work together? I mean, where you have
This.
minimal cyclohexidine and a lot of, neem leaf extract of a formulation of that sort. Would that help in any way?
That is a brilliant idea. We need to check the synergistic effects. Yes,
Yeah,
that's a
that
brilliant
makes
idea. I
much
think we
more
can do that in the future.
Yeah.
That's a very good suggestion. Thank you.
Yeah, thank you.
Yes.
Thank you, Doctor. It's elicited a lot of good, interesting responses from audience, and I think offline communication will progress with this. Thank you very much for your presentation on a very interesting topic.
Thank you.
Now
Thank
we'll
you very
move on to Dr. Azaria Babu. Is he there online? Okay, we'll take next one. Dr. B. Gurdev Singh ji is here. He would be speaking on production and management of quality seeds in NEEM. He is a veteran forest researcher from ICFR Coimbatore, and he's been a very senior scientist, and we're happy to have him for this presentation. Thank you, sir.
So, my experience is with mostly the forestry trees. So, it is about 20-25 years experience which we had with the NEEM. I am going to share with you. And all of you know the importance of the seeds. Next slide. can forward it. Yeah.
You can use this one. This for next.
Okay. Which was?
The down here.
So I am going to speak on, one is the improvement of NEEM, the other one is the NEEM seed collection. So if you see earlier the speakers, my talk is totally a basic work. Actually, any program with the plantations activities has to start with the selection of the good trees. So what type of trees one has to select? So since we were dealing with earlier the timber trees, we know exactly what are the parameters and how these parameters have to be evaluated. So for example, if it is a teak tree, we know that it is a timber tree, we want it has to be straight and it has to be big in size. Whereas in neem tree, what parameters you have to take? Whether you have to see the big size trees, or the trees should have a big crown or large crown, or it should have more number of fruits, or it should have more number of fruit to seeds ratio, or high oil content, or high aza content. How to go about these selections? So that I am going to speak. Likewise, the seed handling, that is from the fruit stage till you use it for chemical extraction or for raising the seedlings, how you have to maintain the quality of the seeds. So these aspects I am going to speak, and I was a part of the International NEEM network. So under the International NEEM network, there are 34 provenance trials were established in 17 countries. In India also we have established about 5 trials, and in our institute control we have established 1 trial. That is a provenance trials. The other big work we did was it in NABARD board project, where about 12 research organizations in India were involved in collection of seeds and estimation of the oil and aza content. So this International NEEM network, these works were done in 1992. To NABARD board work, we did it in somewhere in 1998. And when you say tree improvement, we select the very good trees. Again, the selection, good means what? Like big crown or more fruit yield, those things. And these trees, we can straight away deploy into clone banks and you can multiply them by vegetative propagation and establish a clonal plantations. Or we use these trees, very good trees, and use them for establishment of the orchards. That means we select the best trees and we plant them in one area and we allow them to mate among themselves and try to produce the better quality seeds. Likewise, the collection of the seeds. When you are collecting the seeds, from what source we are collecting the seeds? And neem trees, when you see that, one advantage we have got with the neem tree is has got a very good distribution. So it is there from Uttar Pradesh till the end of the India, that is Kanyakumari, and also from Jodhpur to the Bihar. So that way it is very well distributed, and this distribution makes us to select many good trees. So higher the distribution, greater the scope for the selection, and there will be a lot of variability. So we say the variability is the essence of life, and higher the variability is greater the scope for selections. Likewise, what time the seeds have to be collected, and how much quantity have to be collected, and what method you have to follow, and how to transport these seeds. So selection of the trees for higher fruit yield. So I was mentioning that it should have a good crown, the width of the crown, the height of the crown, we take consider. The another aspect we have seen is the more number of fruit production. So neem trees usually they come into flower in the month of March, April, and the fruits will be available for collection by July, August. And usually if you see this, there will be a flush of leaves in the month of January, February, and flowering will be there in April, and July, August, the fruits will be there. So the fruit formation will be there along with the new branches. So that is how we developed a methodology to count number of fruits which are there per foot length of the branch. In 1 foot length of the branch, how many fruits you are getting? So this is the parameter we took it, and finally we estimate how much fruit each tree is producing. Okay. So for example, this is one tree which we selected, where per foot length we got about 140 fruits per foot length of the branch. So that we consider it is a good fruit yielding tree. The other one is selecting the trees. You have to compare the tree with the comparables. So since in the field we will not be able to make out what is the age of the trees, we take the girth of the trees. So I am selecting a good tree with more number of fruits. I am comparing that tree with the nearby trees, which are of same girth. So that my tree is what I have selected is the best among available trees. So that way in Tamil Nadu and Karnataka and Andhra Pradesh, we selected about 400 trees. So handling these 400 trees was not easy because the fruit is available for a short time. That is in the month of July to August for about 2 to 3 weeks. Within the 2 to 3 weeks, we have to organize the collection and process it. So we recorded all the parameters for the trees, what is its height, the DBH, then fruit weight, seed weight, then germination percentage, oil content, and also aza content estimation. So aza content estimation was done by one of our, oil and aza was done by one of our partner institution. So this is the type of variation we find in one single population. It is not from different regions, it is from the only one region. You see, and when you are collecting the seeds, usually we focus on collecting the seeds from at least minimum 20 trees. So it is a practice that when you collect the seeds from 20 trees from one single population, you are capturing all the variability which is there in that particular population. And you can see that 1, 2, 3, all these things, it is referring to the tree numbers. And you can see the variation in terms of the fruit size. So this is 1 to 10, then we have got 11 to 20 trees. So they are all from the same one place, that is near between Salem and Dharmapuri in Tamil Nadu. So you can see the seeds which we have extracted. If you see the quality of the seeds, the quality is very good. You can see, so they look like almost cashew seeds. So when the seed quality is like this, you will get the maximum output of the azadirachtin. If you have collected fruits which have fallen on the ground and process it, there is likelihood of fungal attack. The germination will be poor, the viability will be less than 3 months. Whereas when you collect the seeds, fruits directly from the trees and process it in a proper way, we can store these seeds for about 1 year. Even after 1 year, you will get about 60% germination. So that is why the selection of the trees is important, and also the processing of the seeds is also very, very important. You can see the variation which you are finding, so much of variation. So when such variation is there in the fruit and also in the seed, there is likely to be variation in the oil and aza content also. So this is the seed physical parameter. Earlier what I have shown was the tree, whereas here it is seed. What is its length and what is its breadth, what is its perimeter, and all those things. So each and every aspect of the seed characteristics have been recorded. And variation in fruit weight, you can see that the weight of the fruits in terms of grams, okay, this is 100 fruit weights, okay. Likewise, the seed weight. So you can see that there is very few trees are with very few seeds with less weight and very seeds with few seeds with more weight, but there will be a average weight, there will be always more. So that is the general principle. So likewise, the azadactin content also you can see this, and it varies from something like, in our total number of trees we have seen, it is varying from about 2,600 ppm to about 16,000 ppm. So this is one population in Tamil Nadu, Kalangal near Karur. We saw that all the trees in that particular population, the aza content was more than 10,000 ppm. So what is the importance of such trees? So you assemble such trees in one place, and when you get the seeds from such trees, again you will get a much higher aza content. The other part is, if you are doing a cell culture where you want to have a start material, if you are starting with the aza content which is very high, your output of the aza content, the total azadactin production will be very, very high. You can have a started with about 4,000 ppm. But if you have got the trees which are about 12,000 ppm, imagine you get 3 times more yield. So likewise, the oil content estimation also has been done. And this is the variation between different populations. And I have shown you the photographs earlier also. There is a variation within the population and between the population also. So that is the advantage in NEEM. Even within a given area also, if you see one tree and the next tree, there will be a difference. So this is the Kalangal population which I mentioned. You can see that this is the record. This is done by Tata Energy Research Institute, aza content. You can see that 10,000 ppm, 12,000, it is almost average is more than 10,000 ppm, whereas national average is only 6,000 ppm. When you see the national average, it is only 6,000 ppm, but there are certain populations where azadactin content is very, very high. This we have done not for 1 year, we have done it for 2 years, just to see is there any difference between 1 year to another year. There is slight difference in the quantity, but the ranking of the trees remains the same. So that is the type of work we have done. The other one is the handling of the NEEM seeds, how to collect the seeds, how to process them. So I mentioned that it will flower in April, and fruits will be ready in July, August. And you will find the fruits invariably, they don't mature at the same time. So you will have a gradation of the fruits, green to yellow, okay? And collection becomes difficult that way, right? So what we have done is we have developed a method that we select these trees and we always harvest the whole fruits, which are yellowish as well as greenish yellow and greenish. And we bring these fruits, and this is an odd photo just to show that how the local people are collecting and processing. Such seeds, they will have a very short viability, and the aza content will also be, yield will also be very less. And this is how the people they are going for collection of the seeds with pole cutters and climbing the trees and collecting the fruits. So different methods of collection. So this is, you can see that the gunny bags in which we collected the fruits. The problem was, it is okay that when you collect the seeds, because we are based in Coimbatore, in Tamil Nadu, we can move the seeds. But collecting the seeds from Andhra Pradesh and moving it to again, processing, that was difficult. So what we used to do is we used to process the fruits in the same place. If you are in Hyderabad, do the processing there itself. Wait for 2 days for them to ripe and do the depulping. So this is the different steps we follow: pre-cleaning, curing, extraction, separation, drying, cleaning, grading, and control of moisture content. So the collected fruits we used to just put in under the shade and allow them to ripe. So I told you that when you are collecting the fruits, they are yellowish fruits as well as greenish yellow and green fruits. So all those fruits will ripen within 2 to 3 days time. So after that, we extract the seeds. So extraction of the seeds is done by rubbing the fruits and brushing them against the baskets. Thoroughly wash it, and we also use, for removing the pulp, we are using the fine sand. Okay, and wherever facilities are there, if there are river side or stream side, do the processing of the fruits there itself. So if you do it processing quickly, the seeds, the damage will be less to the seeds. So this is the seeds which have been extracted from the fruits. Then you can see that we are drying those seeds. So here I told you about 400 trees we estimated, and this is how we process the seeds, and this is the quality of the seeds. And since the seeds will have a high moisture content, so we use a cloth bag to transport and also put it in a cotton box. There also we make the holes, and these are the seeds which is ready for dispatch to other participating institutions. And I told you we conducted the training program for the member countries. So we had about 13 participants from 13 countries. We gave them the training, how to select the good trees, how to process them, and how to clean them, and how to dispatch those trees. This is the part of the training program. And this is the province trial which have been established. 36 trials have been established. In India, we have got 5 trials which have been established. This is the view of this, one of the provenance trials. It's a bit old photo. Now the trees have grown up very well. So what you are seeing here is we got the material from Thailand, and this is the fruits. So they are like almost like grapes. The bunches are very crowded, and the flowering phenology and fruiting phenology is entirely different. This Thailand material were coming into flowering in the month of January, and fruits are available in April, whereas the local material flowering in the month of April and fruits is available in July, August. So if you have got such germplasm, you can also have the fruits in April. So that is the advantage. And we have supplied a lot of these seedlings to the farmers. So this is about raising the seedlings, how to raise the seedlings, broadcasting and transplanting the seedlings into the poly bags. And finally, this is the nursery. We have supplied lakhs of seedlings under different projects. These are the seedlings ready for planting, and transportation of the seedlings for planting site, planting, and watering immediately after planting. Watering is being done, and This is about how to select the good trees, how to process the seeds, and finally how to raise the nursery. So this was the very basic work, and I have shared my experience with you. Thank you.
Thank you, Dr. Gurdev Singh. Thank you very much for your long experience in handling tree crops and particularly neem. And we're very happy to see how systematically the neem seeds are collected and packed and sent to processing. Any quick questions are there on this? Yes, Dr. Ramasamy.
I have a few clarifications from Dr. Gurudev. See, when you say high azadirachtin content or medium azadirachtin content, is it tree dependent or soil dependent or irrigation dependent? When you say azadirachtin, are you talking in terms of azadirachtin A or azadirachtin A plus B? And generally, in my observation, the trees are, giving fruits in an indeterminate space. You can't, collect them at a single space. You have to harvest at different locations. But organized seed collection is almost nil. And I looked at some of the plantations raised by individual farmers and some companies. These trees are not yielding as the trees which are naturally born by bird dropping and so on. Those trees, even I have about 200 trees in my farm, they are yielding very well. And I looked at, the aza content in them, they are above 0.4, 0.5. They are naturally born trees. Nobody nurtured it, nobody put water, and it is randomly grown in my farm in paddy fields and, in the bunds and so on. So my, question is whether organized tree plantation by seeds harvested from different sources claiming to be high azadirachtin content will give you better progeny, or we have to raise from different accessions and choose the seedlings by certain markers will give you a good progeny.
Sir, first of all, I am a person with botany background. I do not know much about azadirachtin content. I told you the azadirachtin estimation was done by Dr. Nutan Kaushik from Tata Energy Research Institute. That information only I have shared. One. Second thing is, the azadirachtin content, our observation is, it is specific to the populations. It is specific to the regions. So, for example, Kalangal is nearer to the Karur. Kalangal is all the trees you have seen, all the 20 trees, almost more than 10,000 ppm. So, I would say that it is the particular area or region specific that may be influenced by the temperature, rainfall and soil factors. But the data what you have shown is for 2 years consistently same type of results we got. So that's what I would say.
No, there is chances of variability and highly, confusing to arrive at a meaningful statement saying that as a high content trees and as a low content trees. That is my concern.
Sir, this.
So my question was that within the same tree, do you see a variation in azadirachtin content in the neem seeds that you get? Is there a variation, and if so, to what extent? Same tree.
We have not studied, so.
Yeah, that's why I thought I had got the partial answer when you said that.
Back here, somebody else raised the hands. There, Mr. Patel, very rich neem seed resource person, 12,000 tons per
Can
year.
I ask? I am Chandubhai Patel in Gujarat, farmer, grower neem tree. My question, Gujarat NEEM tree. How many exact direct in percentage our Gujarat tree?
Sir, Gujarat tree, I do not know.
He has not
What
studied
data I have got
much.
is for Tamil Nadu, Andhra Pradesh, and Karnataka.
Plus, Indonesia.
My request, sir, your research in Gujarat NEEM tree, because NTGCF research is very low.
Okay, thank you. He has not studied much
My
anyway.
colleague, Dr. Tarun Kanti is there. He is from Jodhpur Institute. Maybe he has got a presentation to make also.
I will answer this in my presentation.
Okay, thank you very much, sir. Thank you very much for all the. Anybody else? Well, yeah, you. It's a big problem.
So that's why you have seen our quality of the seeds. That is the method we suggest you do it. It is a bit expensive method.
Expensive method. Okay. Thank you very much. Oh, sorry. Yes, sir.
Good afternoon. I am Dr. RPS Yadav from IFCO. I want to ask, you have selected the tree which they are producing high quality of azactine and neem. So when you collected the seed and raised the saplings, so whether the same quantity of azactine and oil content contains in the seeds of new progeny, or is it difference?
Sir, the collections what we have sent it to Terry is for 2 consecutive years.
No, that is the only tree, identified tree only. No?
Yes.
I'm asking the seedlings raised by the collection of seed. So
sir,
the same
yeah,
things are happening in these trees also.
yeah. So this is now, that is what I was discussing with my other colleagues also. Dr. Rekha Warrior is there, she will talk about that.
No, we will also make a presentation because we have also developed varieties.
So that is very important, whether the high yielding trees will give the progenies which are also going to give the high yield.
Okay, thank you.
Sir, here I would like to add to what Dr. Gurudev Singh has mentioned. I am Dr. Rekha Warrior. I have worked with Dr. Gurudev Singh for 20 years. So here, what Singh sir was mentioning about the Kalangal population. So he said 2 consecutive years. After that, there were quite a lot of people who showed interest in that material. They kept coming back to us asking for that material for various research purposes, like raising of seedlings and for aza content, for cell culture work also. So what we observed, the feedback that we got from them was the amount of azadirachtin that is present in that population is consistently high. And many of the farmers in that area did not realize the value of that material. Many of them have cut down the trees. Now we are trying to encourage the farmers to maintain the trees so that we do not lose that particular genetic resource. It is not that very easy to get trees of that high aza content. So now in the project which Dr. Tarun Kant would be explaining, so there we have tried to support the farmers who are growing these trees, who are rather maintaining these trees on their farmlands. We have taken material from that. We have raised progeny trials. Now those trials have started yielding. We will be assessing the oil and aza of those progenies, and we would be able to tell whether there is a link between what was there in the mother tree and what is there in the progeny. So maybe in a year or 2 we should be able to come out with those results. Thank you.
Thank you. We request now Dr. RPS Yadav to come on the
Thank you,
stage
sir.
and we'll give a memento for our distinguished speaker. Dr. Yadav Sahab. Memento, Dr. Yadav ji. Good afternoon. Now we have a distinguished speaker, Dr. Azaria Babu, formerly director of Tea Research Institute, Toklai, and currently advisor, Upasi United Planters Association, South India. He's going to speak on NEEM, a multifaceted tool in integrated pest management, IPM, for sustainable tea cultivation. Thank you very much, Dr. Azaria Babu, for taking this important subject where a lot of export of tea happens, and MRL is a big issue. Thanks. I hope this will help us to produce a good quality tea. Thank you. Over to you, sir.
Yeah. Thank you. I take this opportunity to thank the organizers, and I regret my inability to present in person. But thank you for the opportunity and giving a chance to speak through the online. And my topic will be the NEEM, a potential multifaceted tool in integrated pest management of tea crop. So coming to the introduction, the tea, I was working for the tea from my, 25 to 26 years in tea. And as a plant protection man, I would like to highlight my experience. The tea is a foliage crop. It has been cultivated as a monoculture plantation crop, typically in the tropical zones. And approximately 20% of the share of the finished product, either in the form of the CTC black tea, orthodox, and the green tea being exported to the developed countries. So this slide shows the tea growing areas in India. It is 50% of the area come from the northeast Assam, 25% from West Bengal, and other 25% rest of the country. So the tea scenario in India, if you take, the All India recorded a production of 1,284.78 million kg in 2024, in which the North India contributed around 82.3% and the South Indian contribution is 17.7. The total area under tea in India is around 6.35 lakh hectare, with 83% of in North India and 16.2% in South India, which covers around approximately 1 lakh hectare of cultivated tea. The South India produced in 2024 226.8 million kgs. So coming to the pest attacking the tea, a variety of herbivores are being attacked from the top foliage to the root system. Then Indian tea industry acquires a crop loss of 20 to 35% despite using the plant protection formulations. So these are all the important pests of tea mosquito bug, the tea thrips, the tea green fly, the red spider mite, the leafhopper or Hypocetra, which is prevalent more in this northeast India. And this slide shows the economic threshold level of the major pests I have mentioned, the tea mosquito bug, aphids, thrips, jassids, and looper, and their ETL is given in the right-hand side. So the economic injury level indicates, based on that we have worked out the economic injury level, so that we need to take appropriate control measures by not allowing the pest to go beyond the economic threshold level. The pest management tea using synthetic pesticide, there are problems associated with the use of pesticide, the residues in tea, resurgence and outbreak of pest, development of resistance, hazards of toxic effect to human and non-target beneficial organisms. So, use of biologicals of plant origin, neem-based bioproducts are used in many crops and their use is very much limited in tea. Neem as a rich resource of bioactive organic constituents. So it's a wonder plant which possesses several bioactive compounds like nimbloid, nimbidin, nimbin, and niminoids are the key active ingredients is the azadirachtin. The effective active ingredient azadirachtin acts as a multi-phase insecticide like repellent, antifidant, growth regulator, and sterilizer. So it controls the pest in storage crops as well as in the plantation crops. The mode of action will be the chitin inhibition of chitin formation and disrupting metamorphosis. Or NEEM is the only sustainable botanical pesticide against more than 300 arthropod pests in different crops. And the major advantage is that unlike the chemical insecticide, NEEM disrupts the endocrine system in insects rather than their nerves, preventing resistant development buildup. So these are all I will be talking about the different effect of this NEEM on the major tea pests like red spider mite, green plant hopper, tea thrips, tea looper, and tea mosquito bug. So the neem kernel aqueous extract, actually this particular technology was developed by RENPAP, and we have run 3 phases of this project through the Ministry of UNIDO supported project through the Ministry of Chemicals and Fertilizers. And this is the method what we have adopted, how to extract the neem kernel aqueous extract from the neem kernels obtained from the field. So 20 kg of neem kernel powder, an adjuvant was added, soak in 100 liters of water, extract and filter through the claw, and add 300 liters of water to make a volume of 400 liters to spray in 1 hectare area under tea. So coming to the activity, the acaricidal activity, when you can see that when we apply the neem kernel powder along with the soap nut, and different treatments are given, 24 hours, 48 hours, and 72 hours, you can see that neem kernel powder with 2.5% and organic synergies 2.5%, that the treatment 3 showed around 90% mortality, which is at par with the normal synthetic chemicals, the hexythiazoles, under laboratory conditions. So the ovicidal of neem kernel aqueous extract is mentioned here. Here also, the egg-killing effect of the neem kernel extract is 90%, which is better than the chemicals. So ovicidal activity, if you treat the different treatments of 5%, 2.5%, and the synergist, we found that within 7 days, the eggs, around 90% killing was noticed. And based on that, we have done large-scale field trials on the evaluation of NKE against the red spider mite, and you can see that the azadirachtin, we have given 2 sprays, and the first week, after second week, third week, total, the average control was around 60% under the field condition. And it is comparable, the neem kernel aqueous extract with the synergist is comparable with the azadirachtin formulation, which is commercially available in the market, 5%. So efficacy of different commercial neem formulation also tested against the different mite, the azadirachtin and the Pon-neem, Linopro, and the NKE. And among the commercial formulation, it is at par and it is effective, and you can make it under the garden level and spray it. So bioefficacy of formulation neem 300 ppm against the tea looper, we found that it is not that much effective. The particular formulation was not, because all the pests cannot be controlled by the neem. And this is the maximum of, we are getting around 13 to 33% mortality. So, but when you use the neem, another formulation, that the antioxidant activity, you can see that the treated leaves are not even touched by the insect, that particular formulation. And based on that, we found out that the growth inhibitory effect of neem as a botanical pesticide against a looper. On the top, you can see that normal larva, pupa, and the adults, when it is treated with the Pon-neem, that deformity characterized by structural defects, stunted growth, and prolonged larval duration, failure of fully to shed their cuticle, and malformed adults are also formed because of the treatment. So bioefficacy of the bioformulation ASA along with the synergist pyrethrum against the Hypocitra, you can see that earlier I told you that it is giving not a good control of the formulations, but this, when it is mixed with the natural pyrethrum, we are getting 100% mortality within 24 hours. Similarly, the green hopper also around 93% control is achieved. So the efficacy of comparison within 300 ppm against the tea mosquito bug, in this case, as I told you, among the different tea pests, tea mosquito bug is the very difficult insect to control, being a sucking pest. And we never find that it is able to control even the available formulations. However, the working on a azadirachtin pyrethrum combination, we got around 70% control, which can be commercially we can formulate it. So we found that there are a lot of natural enemies in the tea ecosystem attacking our pests, and we found that the neem kernel aqueous extract was safer to the predators in the tea ecosystem. And apart from that, the neem kernel extract showed some phytotonic effect. Apart from controlling the pest, you can see the phytotonic effect after 7 days, 14 days, and after a month, that the change is happening because of the phytotonic effect of the 5% neem kernel aqueous extract. Now we have included this as a recommendation in our schedule. So we also do see, when we did the project, we wanted to know that what will we do with that waste after taking the was worked out, and you can see P2O5 and SO4 varies from fresh neem kernel powder and after extraction. So it contains a lot of nutrients, the waste, neem kernel waste. So nutritive value of the waste, after we started composting it to make it as the organic fertilizer. After 30 days and 60 days and 90 days, you can see that there is no much variation in the organic carbon and nitrogen, and it could be used as along with the vermicompost after the 90 days of composting. So we, based on our work done on the neem and the neem kernel extract and the formulations, these are all the relevant publication in the tea pest management and use of extracts. And the role of prosopis, one reed plant, is found to be a very good botanical synergist. When it is added with the neem kernel powder, it enhances the efficacy. This is the growth inhibitory effect upon neem, which is published. And in conclusion, neem acts as a powerful and eco-friendly biopesticide by disrupting the insect growth, repelling the pest, inhibiting the feeding, and deterring the egg laying. Primarily, the azadirachtin making it effective against the mites, thrips, jassids, and lupus without harming the beneficial insects. The supporting the integrated pest management and reducing the reliance on synthetic chemicals in the ecosystem. It works by interfering with the insect hormones, preventing the normal development, and safer to the consumers and the environment. So I thank the World NEEM organization for giving me the opportunity, and all the participants for your patient listening. Thank you.
Thank you, Dr. Azaria Babu, for a wonderful insight into role of neem-based biopesticide in control of tea pests. I'm sure a lot of tea planters would be happy to see. But one question, if you're hearing, for a large plantation, how practical it is to use neem kernel aqueous extract? How many kg of neem kernel required per hectare to make a liquid extract?
Yeah. I will tell you, sir. Actually, when we started this thing, that project was the better way of collection, as the previous speaker told about the neem seeds from the tree, which is not falling and falling on the ground. The seeds were collected and we made the extract. We started with 1%, 1.5%, 2.5%, and 5% neem kernel aqueous extract. Of course, what you told that quantum required for the application is around 22 kg per hectare in 400 liters of water. But by adding the synergist, we found that it can be reduced to 50%. However, as there is no organic control permitted in the tea, when the chemicals are not, the same pests are coming in the tea ecosystem, and which can be managed, and the organic tea fetches a good price, so we have to adjust to that. And some of the NEEM formulation also can be used. Now it is, and if you see that plant protection code released by the Tea Board of India, and in the Central Insecticide Board also, NEEM is the only formulation, biological formulation available for use against the pests.
Thank you, Doctor. Any other questions? Yes, Doctor. From PJ Margo.
How you have prepared pyrethrum extract?
Bio?
How you have prepared pyrethrum extract?
No, pyrethrum, it was a joint venture with the company. They have natural pyrethrum was added in different proportions, and that formulation was initially tested against these pests. Because I know that it is not available in India, so they could outsource and get it to for a project we have done this study.
And what synergist you mentioned, Doctor? What exactly the synergist?
Yeah, that synergist is nothing actually. That soap nut also, right? Soap nut can also be a synergist. And latest in the Ministry of Chemicals and Fertilizer funded projects, we found out that was patented by the IFPT, Institute of Formulation Technology. It is a prosopis, a plant found in the dry areas. And that powder was collected and it is used at 0.05%. That is widely available.
What was the pyrethrin content in extract, pyrethrum extract?
Pyrethrum content, actually I am not able to tell you rightly because it is a formulation supplied by a manufacturer.
2%.
Okay, thank
2%.
you. Thanks, thank you.
Any other questions, sir? Bioefficacy? No? Thank you very much, Dr. Babu, for your presentation on tea. Thank you very much.
Yeah. Thank you, sir.
Thank you very much. Now we have, which one? Yeah. Dr. Balasubramaniam Vairappan from JIPMER. JIPMER, as you know, is a very prestigious medical institute in Pondicherry. He will speak on nimbolide as novel therapeutic agent in hepatocellular carcinoma and metastasis. Doctor, we are agriculture people. Please pardon us for our pronunciation of this subject. Okay, over to you, Doctor.
Thank you, Chairperson. Good afternoon, ladies and gentlemen. First of all, I would like to thank Dr. Nimla for inviting me for this wonderful meeting. So my talk will be consisting the role of bioactive component of Azadirachta indica nimbolide in regulating the gut-liver axis in hepatocellular carcinoma and altering dysregulated microRNAs in HCC. Also anti-metastatic potential of nimbolide in HCC. Coming to introduction, hepatocellular carcinoma is the leading cause of cancer death. So liver cancer is the third leading cause of cancer related death worldwide, accounting for 7.8% of all cancer death. So more than 80% of liver cancer cases are HCC. So it is highly heterogeneous fatal malignancy associated with high morbidity and mortality with limited therapeutic options. In India, the prevalence of HCC is increasing at an alarming rate, posing a major socio-economic and public health problem. The development of effective therapies for HCC, particularly for advanced stage, is therefore critically important. Gut-liver axis is a key pathophysiological mechanism for hepatic disease severity and HCC pathogenesis. Gut microbiota imbalance has been linked to promoting hepatocellular carcinoma progression in preclinical and clinical studies. Increased intestinal permeability, bacterial translocations, and increasing inflammatory conditions are the key precipitating factors for HCC development. So tight junction or intercellular adhesion molecules are located at the apical and basolateral membrane. So which is composed of several proteins including IZO1, occludin and claudin, which play a fundamental role in maintaining tissue barrier and cellular integrity. Intestinal barrier integrity is primarily comprised of intestinal epithelial cells and is further strengthened by the tight junction protein, which restrict the gut microbiota and its products inside the gut. MicroRNA are stable small non-coding RNAs modulating gene expression at the post-transcriptional level by inhibiting mRNA translation or promoting mRNA degradation, which are act as a oncogene or tumor suppressor. MicroRNA based therapies using either microRNA mimics or inhibitors could be a potential future therapeutic approach in combination with chemotherapeutic drug. So we elicited 3 different microRNAs such as 21, 145 and 221 and some of the target gene expression in HCC. Furthermore, extrahepatic metastasis also noted in hepatocellular carcinoma animal models. So lung is the most common extrahepatic metastatic site in HCC. The spread of HCC to the lungs significantly worsens prognosis associated with advanced disease stage and reduced survival rates. Targeting epithelial mesenchymal transition, inflammatory signaling and extracellular matrix remodeling may represent a promising strategy to suppress a metastatic progression in HCC. A nimbolide, a tetratritripenoid sequestered from the NEEM tree, which has a wide variety of functions, including its reduced proliferation and growth, interfering with the growth factor signaling, inhibit angiogenesis and induction of apoptosis and decreased tumor cell migration, invasion and reduced inflammation and induction of cell cycle arrest. So this has been widely studied in various cancer model. However, the potential anti-cancer effect of nimbolide and its molecular mechanism in HCC remain unidentified. So thus we aim to elucidate the role of nimbolide in altering hepatic tight junction integrity in HCC, examine the role of nimbolide in regulating gut microbiota imbalance, intestinal permeability and bacterial translocation in HCC. Furthermore, we also looked at some of the onco and the tumor suppressor microRNAs in HCC, also investigating the anti-tumor and anti-metastatic potential of nimbolide in HCC. Coming to experimental design, so we used Swiss albino mice, 1-month-old Swiss albino mice, and these mice were injected 100 milligram per kilogram body weight to induce hepatocellular carcinoma, followed by N-nitrosomorpholin, NMOR in drinking water to initiate the hepatocellular carcinoma. So the model is about 28-week model. So after 28 weeks, once the tumor developed observed, then the mice were randomized into 5 different groups. So I missed one group here, which is naive and naive animal treated with nimbolide, HCC animal treated with nimbolide, and one more group, HCC animal treated with doxorubicin, which is a known anti-cancer drug. So to compare the efficacy of nimbolide, we used HCC and nimbolide, HCC plus doxorubicin group. So after 28 weeks, the nimbolide was administered to group 2 and group 4 for further 4 weeks. So after, at the end of the experimental duration of 32 weeks, the animals were sacrificed. Macroscopic examination, the nodular formations were observed by macroscopic examination, and we harvested liver tissue, gut tissues, and lung tissues. And blood, from the blood we measured that alpha fetoprotein. It's a gold standard for hepatocellular carcinoma, and the tissues were used for molecular studies, histology and immunohistochemistry, etc. So coming to statistical analysis, all the data were expressed as mean plus or minus standard error of mean. So one-way ANOVA followed by Tukey's multiple comparisons or Kruskal values test. So p-value less than 0.05 was considered statistically significant, and the software were used were Graphite Prism 11. So coming to results. So this figure shows the body weight changes observed in control and experimental mice. You can see here in HCC mice from week 16, the body weight was declined throughout the experimental durations. On treatment with limbolide at the week 28th week for the next 4 weeks, so the body weight is significantly improved. Furthermore, the liver weight is also significantly decreased in HCC and improved by nimbolide, whereas the liver and body weight ratio increase was further significantly decreased by nimbolide treatment. You can see here that this is the gross morphology appearance, and you can see in HCC group there are multiple nodules formations compared to NEEM. And nimbolide treatment, there is the nodular formation was significantly reduced. And this is histological observation from control nimbolide treated HCC and HCC animal treated with nimbolide. You can see here this HCC treated mice, a typical feature of human HCC, increased nucleus and atypical cells and hyperchromatosia. So which are the key clinical features of human HCC, which are reduced by nimbolide treatment. We also calculated the tumor volume and tumor burden, which are reduced by nimbolide treatment. So further we looked at the hepatic marker enzymes and AFP levels in HCC mice treated with or without nimbolide, and you can see here that AFP levels are a marker of HCC which is significantly elevated in HCC is downregulated by nimbolide. Similarly, the other parameters, hepatic function parameters also are significantly reduced by nimbolide. So furthermore, we also looked at the cell proliferation markers, cyclin D1 CDK4 and glypican 3 and PCNA, also noted in HCC mice. The cyclin D1 and CDK4 increased in HCC mice were significantly reduced by nimbolide, and you can also see that more positivity of glypican 3 in HCC mice, which are significantly reduced by nimbolide, and similarly the PCNA immunohistochemistry results also we observed. In addition, we also looked at ROC1, row associated protein kinase, and MMP9 in naive and control experimental mice. You can see that in HCC there is increased ROC1 gene expression, and nimbolide treatment reduced significantly. The similar findings also observed in MMP9. Furthermore, nimbolide also attenuate hepatic inflammation, and we chosen the key inflammatory signaling pathway, NF-kappa B TLR4 mediated pathway, and you can see that pro-inflammatory cytokine, NF-kappa B and TLR4 increased in HCC mice were downregulated by nimbolide. So in summary, nimbolide treatment in HCC mice reduced body weight, improved liver tumor diameter, tumor burden and tumor volume. We found in here nimbolide treatment to HCC inhibited more than 50% of tumor growth, which is clinically significant. Also nimbolide reduced cell cycle marker, cell proliferation and innovation markers. So reduced tumor markers and hepatic marker enzymes. So next, also we looked at the effect of nimbolide in regulating gut-liver axis in HCC. As I mentioned earlier, we looked at different tight junction proteins, including claudins. There are several isoforms of claudin 1, 2, 5, 4, 7, etc. Also zonula occludens 1 and occludin. So these are primary tight junction protein which preserve the tight junction integrity, and we found that claudin 1 and claudin 5 are significantly increased in HCC. This is the protein expression, gene expression, and this is the protein expression, and both showing the similar findings. Elevated claudin 1 and claudin 5 are downregulated by nimbolide treatment to HCC mice. We also validated these findings in immunohistochemistry analysis. You can see here there is a positivity of claudin 1 and claudin 5 in HCC mice, and these were reduced with the treatment of nimbolide. So further, we looked at the nimbolide effect in the intestinal gut liver gut tissues, and these histological findings shows in intestinal villus, and you can see that nimbolide treatment improves intestinal villus architecture in HCC mice. So what happened in HCC? There is a focal flattening of mucosal microvilli and adhesive and presence of inflammatory cells in the lamina propria, so which is regulated, which is reduced with the treatment of nimbolide to HCC mice. We also looked at the occludin, the other important tight junction protein in the intestinal tissues. You can see here in HCC mice there is no positivity of occludin, whereas naive animal and naive mice treated with nimbolide are showing some positivity, and HCC mice treated with nimbolide also showing some positivity. So this is further validated by Western blotting analysis. You can see here that intestinal occludin levels were significantly reduced in HCC mice, and treatment with nimbolide further significantly increased occludin protein expression. And the similar findings were also observed with Z1 protein expression. So further, we looked at the functional assessment of intestinal permeability by assessing by measuring plasma FITS dextran sulfate, okay, fluorescein isothiocyanate conjugated dextran assay. So these are the gold standard for measuring intestinal permeability, and we found that because once that intestinal breach occurs, this will be coming out from the intestine and showing in the circulation. So we found that increased FITS dextran levels in HCC mice, and nimbolide treatment reduced its levels. So also we looked at some of the bacterial translocation markers in HCC mice treated with nimbolide. That plasma lipopolysaccharide binding protein, and soluble CD14 and procalcitonin. These are the key bacterial translocation markers which are significantly elevated in HCC mice, and those mice treated with nimbolide, the levels were significantly reduced. Further, nimbolide attenuated cell adhesion molecules such as E-cadherin and N-cadherin. These are important biomarker for epithelial mesenchymal transition. So we looked at both mRNA and protein. You can see that E-cadherin levels were significantly reduced in HCC. So nimbolide treatment significantly improved. On the other hand, N-cadherin levels were significantly increased in HCC, and which is downregulated by treatment with nimbolide. In summary, in HCC there will be increased inflammation which causes gut dysbiosis, and also disruption of the tight junction protein. So there will be leakage of gut, so more gut derived toxins are released from the gut, and it's entering into the liver through portal circulation and causes epithelial mesenchymal transition, so that extrahepatic metastasis also occurs in the lung. So nimbolide treatment reduces hepatic inflammation as well as gut dysbiosis, and reverses the progression of HCC in mice. So nimbolide also altered dysregulated microRNAs in HCC. So we looked at the three key microRNAs. These 21 and 221 are well studied, and these were increased, and 145 is significantly decreased in HCC, and nimbolide treatment is reversed this regulation. So also we looked at several different genes, Claudin, ROC1, NF-kappa B, BCL2, etc., and also looked at the correlation of those genes with microRNAs, and so we found microRNA 21 has a positive correlation with Claudin 2 and Claudin 5. Sorry, it's a microRNA 221 has a negative correlation with Claudin 2 and positive correlation with Claudin 1, and 145 has a negative correlation with Claudin 5, ROC1, NF-kappa B and MMP9. So thus we summarize. In 10 NMR induced HCC, there is increased microRNA 21 and 221 and decreased 145, which result in increased ROC1, Claudin 1, Claudin 5, thereby increasing cell migration and invasion. Increased MMP9, E-cadherin, N-cadherin, increases EMT and metastasis, and increased NF-kappa B, TNF-alpha, increased inflammation and increased CDK4 and cyclin D1, increased cell cycle progression. So these are contributing to HCC progression. So nimbolide treatment reduces the signaling parameters and has the inhibitory effect in the progression of HCC. So my last observation that anti-metastatic potential of nimbolide in HCC. You can see here that this is a lung tissue strained with hematoxylin and eosin, and you can see that this is a HCC treated lung. Multifocal nodules are there in the HCC mice compared to NEEM, and nimbolide treatment reduced this nodular formation. Also looked at inflammatory signaling pathway in the lung, and similar findings what we observed in the liver, the same we observed in the lung as well. This is the thyroid transcription factor TTF1, so which is the important marker for anti-metastatic potential, and we found that in the lung the levels were significantly increased, and treatment with the nimbolide the TTF levels were significantly reduced. Also we looked at N-cadherin and E-cadherin in the lung tissue, which is increased, similar findings what we observed in other tissues. So in conclusion, Chairperson and ladies and gentlemen, nimbolide inhibits tumor growth by restoring hepatic tight junction protein expression and inflammation in HCC, ameliorate gut dysbiosis and prevent bacterial translocation and improving intestinal barrier integrity. Also it improves dysregulated microRNAs. Further validation through clinical and experimental studies may support the inclusion of nimbolide as a potential therapeutic strategy for HCC management. I would like to acknowledge our funding bodies and my lab members. Thank you for your kind attention.
Thank you, Dr. Balasubramanian. Wonderful research you have presented, use of NEEM in the medical research. We're curious to know where the nimbolide fraction, what is the source of your raw material for these trials?
Source of nimbolide.
Yeah.
So nimbolide, we come. This is come from Azadirachta indica, the NEEM, but we commercially purchase this active component.
Oh, you bought the active fraction?
Yeah, active fraction.
Is it available for buying as active fraction?
Yeah, it's active principle present in the neem, flower, leaves and seeds, etc.
It gives a good confidence for us to produce and make it available for cancer research. Wonderful, thank you. It's a very value addition for our industry. Thanks for giving this kind of a lead to us.
Thank you.
And JIPMER kind of institute doing the wonderful work. Gives confidence to the entire industry. Any other questions, please? Yes, doctor.
Question. Regarding the route of administration, you showed an injection and are you orally administering it or you are injecting it?
Nimbolide treated with, dissolved with PBS, saline, and injected orally, gavaged.
Okay. My second question is, so after you have subjected the mice for a certain period and you have done your analysis, have you also seen after the withdrawal of nimbolide administration in a cancer model of HCC, has the cancer really relapsed again? Because that's normally the case in most cancers where the disease relapses after the treatment stops. So do you have any model that has?
No, actually we cull all the mice, so at the end of the experimental duration, so none of the mice will be survived.
No, I mean you have administered it for a certain period and
Yeah.
then not
Yeah.
administered, and then after that, say you have administered for 5 days, and you
Doctor,
have
Doctor?
not administered for the next 10 days, and then you sacrificed
So
and done
no,
your
no,
histochemical.
Doctor?
No, we
histopathological
haven't checked
Sorry
like that.
for interruption.
Yes.
Actually, we are running short of time.
Okay, sir.
You can have your discussion offline
Yeah.
with him after the session is over.
Yeah,
Yes.
I'll explain to you. So my experimental
Yeah.
results. So
Yes.
thank you so much.
Thank you very much for the wonderful session. My request to all participants is, it's tea break, but it's running behind the time. Our request is, please go to the tea hall, bring the tea here as early as possible so that we can conclude next session in time. Meanwhile, I request Dr. One second. I request Mr. Pradeep Jaipuria ji, please come on the stage to give a memento for Dr. from Balasubramaniam from JIPMER. I think we can just relax, go, get some chai, tea, and come over here for the next session. 10 minutes time.
We'll reassemble in 10 minutes. Hello?
Reassemble in 10 minutes time.
Excuse me. We will reassemble here in 10 minutes time. Currently it is 4:40. 4:50 we are reassembling.